Hi there, and welcome to this video on GCSE Biology for the AQA Specification, focusing on reproduction and in particular on genetic inheritance. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started. Hello, and welcome to this tutorial on genetic inheritance. So in today's tutorial, we're going to be having a look at some basic definitions, single gene characteristics, and finally coming on to have a look at Punnet squares and what we mean by them. So first of all, let's go through some really important terms that you need to get your head around. So you've all heard of chromosomes, but what actually are they? So they're a structure in the nucleus of a cell that is made up of one condensed molecule of DNA. Okay? So your DNA is found in your chromosomes. Now, a gamete is a sex cell, so we've come across what gametes are in previous tutorials in this lecture series. So if you're not quite sure, perhaps go back and watch those, but I'll explain again just in case anyway. So a gamete cell is a sex cell, and therefore these cells contain half the normal number of chromosomes. So the normal number of chromosomes in a human being is 46, which makes normal cells diploid cells. And that's because they contain pairs of homologous chromosomes, which basically means that within the 23 pairs of chromosomes that there is a maternal and a paternal chromosome in each of those pairs. So, for example, chromosome 1 is a pair, and so there will be a maternal chromosome and a paternal chromosome within that pair. So the maternal chromosome 1 and paternal chromosome 1 make up chromosome 1 pair. So homologous pairs of chromosomes make up diploid cells. Whereas gamete cells contain 23 chromosomes, so half the normal number of chromosomes. And that's because they only contain a single set of chromosomes. The chromosomes do not come in pairs. So this makes gamete cells haploid cells. So this was covered in a previous tutorial. So if that doesn't make sense to you now, go back and watch that tutorial because it will just be explained again, but just in a slightly different way. But all you need to get from this is that gametes are haploid cells, meaning that they contain half the number of chromosomes of normal cells. And sometimes we use this notation. So haploid cells have N chromosomes, diploid cells have 2N. So that's our single set of chromosomes, that's our pairs of homologous chromosomes. So N is equal to 23 here. Diploid cells contain 2N, so 23 times 2, 46 chromosomes. Now, just to look at this in another way, think of the stomach, which has cells like everything else in the body. And a stomach cell is a normal cell. It's not a gamete cell, it's not a sex cell. So therefore it's a diploid cell, which means it must contain 46 chromosomes. Now let's have a look at our gametes. So in females, our gametes are egg cells, whereas in males, their gametes are sperm cells. But either way, both the sperm and egg cells are haploid cells, which means that they contain half the normal number of chromosomes. So they contain 46 divided by 2, which equals 23 chromosomes. And this makes really perfect sense because if you think about it, in fertilisation, the egg and the sperm cell come together. 23 plus 23 equals 46. There we go. That's our normal cell that we need to then allow this baby to establish, right? So that's how I like to think of it. So gametes contain half the normal number of chromosomes because at the end of the day, the gametes come together in fertilisation and add their chromosomes together. Now, a gene is a section of DNA that codes for a certain protein or characteristic, and an allele is just a certain version of this gene. So here's our chromosome again, which contains condensed DNA. Let's zoom in on it, that's our DNA there. Now if we take a section of this DNA, that's our gene, and in this case we're saying that this section of DNA is the gene for eye colour. Now we all know there's variations on eye colour, some people have blue eyes, some people have brown or green eyes. So this variation means that there's a certain version of a gene that codes for the blue eye colour, for example. And another way of saying that, instead of saying all those words that I just said, is an allele of a gene. Okay, so that's all that means. So within the gene for eye colour, there are different alleles. Each allele codes for a different eye colour. Now, a person will have 2 copies of an allele for a given gene. So, for example, everyone has a gene for eye colour. So one person may carry an allele for blue eyes and an allele for brown eyes, whereas another person might carry 2 alleles for brown eyes, as we can see here. So person A, on their eye colour gene, has a blue eye allele and a brown eye allele, whereas person B has 2 brown eye alleles. Okay? So just different versions of the same gene, and you contain 2 of those different versions for each given gene. Now moving on to dominant and recessive alleles. So if we have a certain version of a gene, so an allele, and if that allele is dominant, then that means it is always expressed in an organism. Now, if the allele is recessive, that recessive allele is only expressed when the dominant allele is not there. Now this might be a little bit tricky to get your head around when we just look at it in terms of words, so we're going to have a look at some examples to make it a little bit easier. So for example, we can say brown eyes are a dominant allele and blue eyes are a recessive allele, okay? So we're saying our brown eyes are our dominant allele. So person A has 1 brown eye allele, and person A also has a recessive allele that codes for blue eyes. So that means that because we have this dominant allele, present, it's always going to present. So the dominant allele here is our brown eyes. So that means person A must have brown eyes, even though person A also has the blue eye allele, but it cannot be expressed because the dominant one is always dominant. So now let's look at person B. So we said person B has 2 blue eye alleles, and that's perfect because blue eye alleles are not dominant, they're recessive. So in order for them to be shown on person B, only the blue eye allele has to be present. Nothing else can be, no other dominant allele can be present. So this means that because person B only has recessive alleles with no dominant alleles, person B has blue eyes. So if I just go back to the previous slide, person A had this dominant allele, and therefore the blue eye recessive allele was not expressed because brown eyes rule over blue eyes in this case. But when the brown eye allele was not present, the blue eye allele can show. And it's showing in the phenotype of the person, and we'll come on now to see what that means. So the genotype is the raw genetic sequence of an organism. The phenotype is the physical characteristic expressed by an organism based on the environment and genotype. So how I like to remember this is that the genotype is kind of like the raw code. The phenotype is how it's presented on the person. So the genotype you can't see, but the phenotype you can see. Now make sure you learn these definitions. We've gone through a lot of them, but once you know them, everything will be a breeze and you'll get lots of easy marks in the exam. Now, just to get over what the difference between these words, so the genotype is what we don't see. So the alleles of person A are what we don't see. We can't actually see that person A has this blue eye recessive allele and the brown eye dominant allele, because that's in their genotype, in their genetic sequence. What we do actually see is the dominant allele being presented on person A. So person A has brown eyes because they have the recessive blue eye allele and they have the dominant brown eye allele. So therefore the brown eye allele always presents, and that's what we see in the phenotype. So the phenotype is what person A is presenting to us with, and that's a combination of the genotype and the environment. Whereas the genotype is just referring to the exact alleles that person A has, and we know because previously we saw in a slide that person A has the blue eye recessive allele and the brown eye dominant allele. Okay? So that's the difference between genotype and phenotype. So now let's have a look at red-green colour blindness. So the majority of characteristics require a few different genes in order to be expressed. Some specific characteristics, on the other hand, are controlled by only one gene. So an example of this is the red-green colour blindness gene in humans and fur colour in mice. So these are often used as examples in the exam. Red-green colour blindness, however, is more complicated due to other circumstances, but it is important to remember these two examples as examples of single gene characteristics. So now let's have a look at an example. So the allele for dark hair is dominant, whereas the allele for light hair is recessive. Now if both alleles provided by the parents are dominant, so if the genotype, the alleles are dominant, the person will have dark hair. Now remember, the outcome of the genotype combined with the environmental features is the phenotype. So this is regarded to be homozygous dominant. We say homo because we're referring to only one type of allele because we're saying that both alleles are dominant. It's not one recessive and one dominant allele, it's both are dominant, therefore we say homozygous because both alleles are the same, and the stem homo means the same, or one. Now let's just have a look at this example to make it a little clearer. So person A's alleles for hair colour are the dark allele and the dark allele. So these are both dominant alleles, and they are homozygous because both alleles are the same. So that's why we call person A's alleles homozygous dominant. And person A's actual hair colour, or the phenotype of person A, is dark hair as well, because the person has these two dominant alleles. Now, if both alleles are recessive, the person will have light hair, and this is considered to be homozygous recessive. So, for example, person B's alleles for hair colour are light and light. So, homozygous again, because both alleles are the same, but this time, instead of dominant, we know that these hair colour alleles are recessive. So, therefore, we call person B's genotype homozygous recessive. And what do they present with? Well, because only recessive alleles are present, no dominant alleles, the recessive allele is going to show in the phenotype, and therefore person B has light hair. Now let's have a look at what happens when we have a mix of dominant and recessive alleles. So we call this heterozygous. So the stem hetero means that there's more than one, okay? So this makes sense if you think about it because we have the dominant and recessive. It's not dominant or recessive, it's dominant recessive. So it's mixed, okay? So this works as the dark hair is the dominant allele. And because the dominant allele is present, it's always expressed. So just to put this in another diagrammatic format to make it clearer for you, person C has the dark allele and the light allele. It's heterozygous because the alleles are different. And so remember, the stem hetero refers to them being different, whereas the stem homo refers to the alleles being the same. Now, dominant is because they express the dominant allele. So therefore, person C's actual hair colour is going to be the dark colour, because even though this light colour is present, we have a heterozygous combination. Remember, hetero, that's stem, it means different. The alleles are different, and we know the dark allele is dominant, so that is always going to be the one expressed. In order for the light allele to be expressed, because it's recessive, only the light allele can be present. Okay, so I hope that made sense, but if not, do go back and have a look through it again, because once you work it out and once it clicks in your head, it's really, really easy. Now, with some genes, you can predict the phenotype that is expressed, dependent on the alleles of the parents. So this can be done with single gene crosses. Therefore, these phenotypes must only be influenced by one set of alleles. But when there's more than one set of alleles that influence a phenotype, it's much harder to predict the probability of the characteristics shown. So, just in terms of our notation from now on, in order to show a dominant allele, we use a capital letter, and to show a recessive allele, we use a lowercase letter. So you'll see what I mean by that now. So we'll work through a little example. So we're saying that Dad has dark hair, and Mum has light hair, so blonde hair here. Now, the alleles for the father, we know that Dad has dark hair, so therefore there has to be a dominant allele somewhere in there, because we know the dominant allele is our dark hair allele. And we're going to use the letter D for this circumstance, but any letter can be used in theory. So, because we know it's a dominant allele, it doesn't matter what the second allele is, as long as one of the alleles is a capital D for dominant, okay? So therefore, the father can be homozygous dominant, so 2 dominants, or heterozygous, so 1 dominant, 1 recessive allele. And that's the genotype, okay? So homozygous dominant, where we have a dark hair allele and a dark hair allele, or heterozygous, where we have a, underline it like that, where we have a dark hair allele and a light hair allele, okay? Whereas here we have 2 dark hair alleles. Now these are the options for Dad. Homozygous dominant or heterozygous dominant. And remember I said homo is for when both alleles are the same, hetero is for when the alleles are different. Now Mum has light hair. We know that light hair has a recessive allele, therefore it's only expressed in the phenotype, so in the mother it's only expressed visually when we have 2 recessive alleles. No dominant allele can be present. So that means our only option for Mum is to have 2 recessive alleles, which we refer to as homozygous recessive. And we say homo, not hetero, because homozygous, we have 2 of the same alleles, and they're 2 recessive alleles. Now let's have a look at putting this into a Punnet square. So we're going to say that the father is heterozygous dominant, which means that he has 1 dark hair allele, 1 light hair allele. And then we're going to predict the possible genotypes for the offspring of the mum and dad in the example. So this is what we call a Punnet square. So first what you do is you make your table. Now this box is always going to be left empty. Now you can interchangeably put the father or mother between these 2 spaces, but we're going to put father up here, mother here. Now we know the father's genotype is heterozygous. We know mother's genotype is homozygous recessive. So we split the alleles up, so we put dad's dark hair allele over here and dad's light hair allele over here, and we put mum's light hair allele over here and mum's other light hair allele over here. Okay, so that's what we do first of all. So you have your alleles of your parents here. Now this is going to disappear, I think, when I change slides. But that's okay because you should understand why I've put the letters where they are. So we place the alleles of one parent at the top of the Punnet square and one set of alleles on the side, and then what we do is we write one allele from each parent per box. Okay, so let's say I drop these down here. So all I've done here is I've filled in these boxes with Dad's alleles. So going down in this column, I've written his dark hair allele. And going down here, I've written his light hair allele. And now we're going to add Mum's to these 4 boxes. So simple as we add Mum's light hair alleles into these boxes of the offspring. And this will allow us to predict the alleles because as you can see, we have, let me choose a colour, we have 2 heterozygous. So we have 2 of these. And we have 2 recessive, homozygous recessive. So this is in the offspring. So this is the probability, or it shows the combinations that we may possibly have in the offspring of this father and this specific mother. These are the possible offspring genotypes that they can have. And as you can see, they have, if we write this as a ratio, they have 2 heterozygous and 2 homozygous recessive, which means it's a 1-to-1 ratio, which means the probability of their child having dark hair, so that would mean they'd have to have this combination, is 2/4, or 1 in 1, so 0.5. So the probability of their child having dark hair is 50%. But the probability of their child having light hair is 50% as well. And as we said, the ratio therefore would be 1-to-1. So this technique can be used to work out the proportion of having any characteristic that is a single gene characteristic. So as I've said, our outcome of our offspring are either our heterozygotes, which are going to be big D, little d, and because they have the big D present, they're going to have dark hair, or little d, little d, and because they don't have the dark hair allele present, they're going to have light hair. So make sure you can draw Punnet squares like these, as AQA often provides marks for these. Now that's all for today. Well done. This was quite a meaty tutorial with lots of definitions, so do go back and re-watch. And I've tried to explain this as much as I can, so I really hope that makes sense to you. And I'll see you for the next session. Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.